Hello, my name is Sarah Taylor, and I am doing my presentation on toxoplasma gondii. Let's begin with an overview of this organism. All organisms fall into taxonomic classification, starting from the most broad domain going all the way down to species. Now, as we can see here, the domain is Eukarya, the kingdom Alveolata, phylum Apicomplexa, class Codacea, order Eucodisauria, family Sarcocystidae, genus Toxoplasma, species Toxoplasma gondii. I apologize if I got any of those pronunciations wrong. What is the known habitat for this parasite? So, the only known definitive host for Toxoplasma gondii is members of the Felidae family, aka domestic cats and their relatives. They're the only known hosts. There are also intermediate hosts in nature, which are birds and rodents. They can also be found in contaminated meats, whether that be poultry, it can be cow, pig. Any meats can become contaminated, whether they're on land or they're in water. It can also get into soil, and so therefore it can contaminate your water supply, it can contaminate your plants and vegetables, but this all normally comes from cat feces. So a big habitat for this parasite is within households that don't have who have cats that have Toxoplasma gondii, that it hasn't been treated for, and that can be like found in their litter boxes and such. So that's a big habitat for them. Nutritional requirements. So Toxoplasma gondii is an obligate intracellular parasite. That means it needs to get all of its food sources from the host cell. It lives in the host cell, and so it will hijack the nutrients that come in. And as you can see from this diagram, these ones in red are coming in to these. These blue things are the Toxoplasma gondii in the cell. And so amino acids come in, iron comes in, thymine comes in, polyamines come in, and then through that they're going to get their nutrients. So here's the appearance under a microscope. So this picture here is stained, so this purple stain here is from the GEMA stain. So they're approximately 4 to 8 micrometers long, 2 to 3 micrometers wide, tapered in the interior ends and blunt in the posterior ends, and they have a large nucleus. So this dark blue, that's going to be its really large nucleus. And then let's see, this tapered would be its tapered interior end, and this is, see, it's more wider and sort of blunter. That's going to be its blunt posterior end. Now I will take you through the pathogenesis of this parasite. So as we said before when we were talking about habitat, transmission all has to do with this habitat as well. So as you can see, it's all from these fecal oocysts. There are multiple routes of transmission, but they all come back to things that are contaminated with fecal oocysts. So as you see, this is our primary known host, and these are our intermediate hosts. So cat will expel fecal oocysts, and then they can infect rats, birds, and then they get tissue cysts, which then go back to cats, which then go back to fecal cysts. And then these fecal cysts, whether, it comes from the cat or maybe, the pig or something else eats these contaminated intermediates, they can go up to animals that we humans eat, and then they create tissue cysts, and then we consume this infected meat, and we get infected. Also, fecal cysts, as I said, can get into fruits and veg, our water, our cat's litter trays, and then by not washing our fruits properly, or not treating our water properly, or not wearing gloves when dealing with our cat's litter box, we can get infected. Another way, though highly rarely, another way it can get transmitted is through infected blood. So it can live in the blood, even though it creates tissue cysts, it can travel in the blood, and so therefore in certain stages you can get an infection from contaminated blood. It's also possible here for a mum to be infected and have tissue cysts, and this can in cases confer to an unborn fetus. So, host invasion. Parasites form tissue cysts in commonly the skeletal muscle, the myocardium, the brain, and the eyes. And what this picture is showing here is a tissue cyst within brain matter. So you can get these cysts in your brain, and they can be quite concerning, and they need to be taken care of. Just like they can get in your eyes and cause damage, especially if it's involved with your retina. It can cause problems in your muscle. It can cause problems in your myocardium, which causes problems with your heart. So how do they do this? So, mechanism for these parasites getting in. So they do something called active invasion, and here is a picture showing that. So you're going to have this parasite right here. It's going to form a nice tight junction, like bonding with the plasma membrane of the host cell it wants to get in. And so, see right here, forming its tight junction. It's then going to form a moving junction. And what this does is allows the parasite to pull itself into this cell, just straight forcing its way in, all the way till it's inside. And then the moving junction stops. It just disappears. Important virulence factors that help, these parasites stay alive and survive within host tissues. A big one is ROP18 or Rospidase 18. So what this does is it causes phosphorylation of several IRGs, which causes reduced IRG recruitment to the PVM. What this results in is persistence of the parasite in an activated macrophage. So if a macrophage has been presented the parasite's tissue and is even hunting for it, even if it's activated, it can still live in that macrophage due to this component. So here are the symptoms of toxoplasma gondii. So most cases are usually non-severe. A lot of us by now know the term asymptomatic. Most people, especially non-immunocompromised people, are asymptomatic. Sometimes people can experience flu-like symptoms, so that's your swollen lymph glands, muscle aches and pains. Other symptoms are really what come with ocular toxoplasmosis, which is an infection into the eye. So that can get reduced vision, blurred vision, pain, often with bright light, so light sensitivity, redness of the eye, sometimes tearing. And this can, if this gets incorporates into the retina, can cause some lasting damage. Alright, now I'm going to take you through the epidemiology of toxoplasma gondii. So who is at risk for getting this. Everyone can get this. Unfortunately, it can live in our cats, our pets, and so that is a big infection point. And because of that, and because such a large population has cats, everyone is susceptible to it. Also, in places where they have like mountain lions and such, it can be in the soil. So, to reduce risk, you want to cook your meats to a safe temperature that allows the parasites to be killed, so they're no longer, while they might still be in the meat, they are no longer alive and they can't infect you when you consume it. Don't drink untreated water. If you're out hiking, boil your water. That will kill it. Don't just chemically treat it. Wear gloves when dealing with soil or sand that might be contaminated with your cat feces. So, if your cat plays around in the backyard when you're doing gardening and stuff, wear gloves. Once you've come in from playing in the backyard, wash your hands. When you're dealing with a cat litter, gloves, wash your hands. And then, of course, wash your fruits, vegetables, and meat thoroughly, and anything you use to prepare it, because the parasite can move on to different surfaces. If it's like on the surface of your fruit, you put it down on a bench, it can move to that surface. So just make sure you wash everything thoroughly and clean everything up when preparing food. And the biggest thing, of course, is to wash your hands extremely thoroughly. Now, what circumstances are riskier for people when they contract toxoplasma gondii? So someone who is pregnant can have, while they are normally pretty healthy, the pregnant people also can be in an immunocompromised state, and at time and usually are a bit more immunocompromised than when they're not pregnant. But this can also affect the fetus growing inside them, and this can cause miscarriage, a stillborn child, or a child born with signs of congenital toxoplasmosis. Congenital just meaning they contracted it from their mother. And so they can come out with signs like that. So of course, someone who's pregnant needs to be more careful. Getting ocular toxoplasmosis is also much more riskier because it can cause progressive blindness if central structures in the retina are involved. And then of course, anyone with a compromised immune system is more at risk, more at risk to have severe symptoms and more likely to have a relapse of an infection. So if you got infected when you were healthy, and then say, you got pregnant and you're immunocompromised now, you could have a relapse of that infection. Or say you got infected when you were healthy and then you develop something like HIV or AIDS, that can cause a relapse of an infection. Or even someone who becomes infected as an immunocompromised person can have a relapse later down the track, because the parasite can stay in a latent stage in your tissue and reemerge when they see that your body's a bit weaker than before. Prevention. So how can we prevent this? Diagnosement. So how do we diagnose? We diagnose through serological testing. So mainly this is measuring IgG, immunoglobulin G, which is an antibody type. If we need to figure out time since infection, which can be really important for pregnant people, we can look at IgM, which is the first antibody type that shows up in an infection. You can also do direct observation of parasites in the stained tissue section, CSF, which is cerebrospinal fluid, and other biopsy materials. They can be put on a slide, they can be stained, and you can physically see under a microscope those tissue cysts. The parasites can also be isolated from the blood or other body fluids, again CSF, cerebrospinal fluid, and you can isolate them from that fluid and you can see them. So treatment for this illness. So for most people, this does not need to be treated. Most people are going to be asymptomatic. They're going to feel a bit achy, maybe if they get symptoms, those flu-like symptoms, but they're not necessarily going to need treatment. However, infants, newborns, pregnant people, and immunocompromised people who all have, infants, newborns, and immunocompromised people all don't have the greatest of an immune system, so they can be treated, and then pregnant people can also be treated so that a fetus doesn't contract it. And it's normally treated with a combination of drugs. The most common combination is primethylamine, sulfadiazine, and folinic acid. This is the most common drug combination to treat this. So drug susceptibility, you know, what drugs work really well for toxoplasmosis and what don't work well at all. Um, so toxoplasmosis, as we talked about in the last slide, is mainly treated by pyrimethylamine and sulfadiazine, and these are drugs that Toxoplasmosis gondii is widely susceptible to. However, there have been cases where this treatment does fail. Um, Toxoplasma gondii does have a very high resistance in some mutations to erythromycin, spiromycin, and synthiamycin, which is this resistance is thought to be encoded in the rRNA genes. So let's talk about drug resistance and how this possibly works within the parasite, and what drug resistances we do know. So the setting. There isn't a lot of data about, where antibiotic resistance per methylmelanin is. It's not widely found within different strains of Toxoplasma gondii, um, but it does happen. Permethylene resistance, as shown in papers who have done single-point mutagenesis, have shown that resistance can come from three possible sources in the DHFR-TS gene. And so this is the three mutations are a single point mutation at amino residue 25 from W to R, seems to convey permethylmethylene resistance. Single point mutation at amino residue 98 from L to S shows resistance to primethylmethylene resistance, and then at amino as residue 134, L to H can also show primethylmethylene resistance. Unfortunately, we do not know at the current time the mechanism that works to convey this resistance to this drug. We only know that it can happen, it does happen, and that in laboratory testing, when they did these 3-point mutations, just a single point mutation, these three showed, if when it was just that one changed, that it did convey some resistance. Here are my citations for all the articles and websites I used in the creation of this research.